Hey guys, so the next unit that we're going to talk about is blood. So some of the stuff you might have heard before, but others hopefully you're hearing for the first time. So what makes up our blood? Our blood is made up of red blood cells, which are called erythrocytes. These are the most abundant cells in our blood. They are produced in the bone marrow and contain a protein called hemoglobin that carries oxygen to our cells. White blood cells, which are called leukocytes, they are part of the immune system and destroy infectious agents called pathogens. And plasma. This is the yellowish liquid portion of the blood that contains electrolytes, nutrients, vitamins, hormones, clotting factors, and proteins such as antibodies to fight infection. And then platelets, which are also called thrombocytes. These are the clotting factors that are carried in the plasma. They clot together in a process called coagulation to seal a wound and prevent a loss of blood. So the average adult has about 5 liters of blood inside our body. Basically makes up about 7 to 8% of the body weight, give or take. Blood is a living tissue that carries oxygen and nutrients to all parts of the body and carries carbon dioxide and other waste products back to the lungs, kidneys, and liver for disposal. It also fights infection and helps heal wounds so we can stay healthy. There are about 1 billion red blood cells in 2 to 3 drops of blood. For every 600 red blood cells, there are about 40 platelets and 1 white cell. So what does the abbreviation BPA stand for? So that stands for blood stain pattern analysis. What can an investigator learn from the analysis of blood spatter? Hopefully you did some of this last year or last semester. It can tell you the type and velocity of weapon, the number of blows, the handedness of the assailant, right or left hand, position and movements of the victim and assailant during and after the attack, which wounds were inflicted first, type of injuries, how long ago the crime was committed, and whether death was immediate or delayed. So lots of different information that you can get from blood stain pattern analysis. So how is blood evidence detected at a crime scene? So first, we have light source. Investigators will first examine the crime scene to look for areas that may contain blood. They may use a high-intensity light or UV lights to help them find traces of blood as well as other bodily fluids that are not visible under normal lighting conditions. And then they can also do blood reagent tests. These tests, referred to as presumptive tests, are used to detect blood at crime scenes based upon the properties of hemoglobin in the blood. Further tests at the crime lab can determine if it's human blood or something else. Examples would be phenolphthalein, which is a chemical that is utilized today, usually referred to as the Kastle-Meyer test, and produces a pink color when it reacts with hemoglobin. And then Hemastix is a strip that has been coated with tetramethylbenzidine, otherwise known as TMB, and will produce a green or blue-green color with the presence of hemoglobin. Luminol can also be used, very widely used actually. This chemical is used by crime scene investigators to locate traces of blood, even if it has been cleaned or removed. So this is very useful in trying to see where people have tried to cover up the fact that something happened there. Investigators spray a luminol solution throughout the area under investigation and look for reactions with the iron present in the blood, which causes a blue luminescence. One problem is that other substances also react, such as some metals, paints, cleaning products, and plant materials. Another problem is that the chemical reaction can destroy other evidence in the crime scene. So you have to kind of be careful of your timing and stuff like that. LCV, or leucocrystal violet, is one type of chemical process that is used for blood enhancement. Using this test helps to make the blood evidence more visible so it can be photographed and analyzed. And then fluorescein. Fluorescein is a chemical that is capable of detecting latent or old blood. Similar to luminol, it is ideal for stains or smears found throughout the crime scene. So this is just giving you some different blood stain patterns. We have spatter, which is created from the application of force to the area where the blood originated. Origin is the place where it came from. Angle of impact is the angle at which the blood droplet strikes a surface. The parent drop is the droplet from which a satellite or a spatter originates, like you see over there on the picture. Satellites are the small drops of blood that break off from the parent. And spines are the pointed edges of the stain that radiate from the spatter. That can help determine the direction from which the blood traveled. Passive blood stains are patterns created from the force of gravity, drops, series of drops, flow patterns, blood pools, stuff like that. Projected blood stains are patterns that occur when a force is applied to a source of blood. Includes low, medium, or high impact spatters, cast off, arterial spurting, expiratory blood blown out the nose, mouth, or a wound. And then you have transfer or contact blood stains. These are created when wet, bloody objects come in contact with the target surface, may be used to identify an object or body part. A white pattern is created from an object moving through a blood stain, while a swipe pattern is created from an object leaving a blood stain. So your blood type is established before you are born by specific genes inherited from your parents. You get those from your mother and your father. Determine your blood type by causing proteins called agglutinogens to exist on the surface of all your red blood cells. So there are 3 alleles. Basically, you have an A, a B, and an O. So up in the top right here, it shows you the way you can get type A, then the way you can get type B, and here's the way you can get type O or type AB. If you remember some of your genetics, A and B are co-dominant. That means they both show if you have type AB, and both A and B are dominant to type O. So if you have an A or a B along with an O, then you just end up with A or B. So this is just a chart showing you, that you could fill out saying how you could get the different things. A blood transfusion is a procedure in which blood is given to a patient through IV line in one of the blood vessels. Blood transfusions are done to replace blood loss during surgery or a serious injury. Transfusion also may be done if a person's body can't make blood properly because of an illness. So who can you give blood to? It depends on who you have. Type O are the universal donors. They can give to anyone. Type AB are the universal recipients because they can get blood from anybody. So they're the lucky ones. And then there's the RH plus or RH negative. That's the rhesus factor. RH plus can receive either. RH minus can only receive negative. And this is just showing you like how many people, what percentage of the people have. So I am B positive. So that means I fall in a group with 9.4% of other people. The RH factors were studied in the rhesus monkey to learn about human anatomy, and that's the plus or minus. So the presence of the protein or lack of it is referred to as the RH factor or rhesus factor. If your blood does contain the protein, then you are RH positive. If it does not, then you are RH negative. And here are just some microscopic views of different blood cells. This you can pause and go through this if you would like. These are just genetics basics. Genotype versus phenotype. And that's it.